So we are going to get started on our afternoon session. So I'll pass the mic over to Anu.
Aloha mai. Can you guys hear me good? Good? Okay, good. So for the final session today, we'll be transitioning back to student presentations, which will run until about 4:40. As a reminder to presenters, without automatically advancing slides, please just say next slide or give an obvious nod and the moderator will advance the slide. And so first up, we have Ryan Liu with the presentation entitled Human in the Loop AI to Advance Tropical Science Annotation and Data Collection. And just want to make sure that Ryan is in the panelist room.
Hello? Oh, can you hear me?
Yeah, perfect.
Okay. So I'll just say next when I want to move to the next slide.
Sounds good. I'll hand it off to you, Ryan.
All right. Hello, everyone. My name is Ryan Liu, and I am currently interning for Professor Travis Mandel this semester. This internship is researching applications for human-in-the-loop AI for environmental science problems. Next. Oh, okay. Artificial intelligence and machine learning are revolutionizing the way scientific data is analyzed. AI can reduce the amount of human effort involved by automatically finding and exploiting patterns in the data set. Unfortunately, there are still areas that are done completely by people with no AI involvement. There is still a lot of human effort involved in the earlier steps in the pipeline on the right, specifically data collection and annotation. Is there a way we can incorporate AI and machine learning in these steps as well? Next. So I know that most of the audience are not researching AI and machine learning. Why should biologists and environmental scientists care? There are several areas where TCBES could use or are already using AI in their research. For example, in detecting coral or finding birdsong patterns. For people in TCBES, how much time do you spend on collecting or annotating data? Our goal is to incorporate AI and machine learning to make this process easier for you. Next. Human-in-the-loop AI research is currently focused on two projects. We have the the interactive annotator and the fish tracker. Next. As I initially explained, machine learning is traditionally involved in learning from annotated data to produce future labels and predictions. How can we push this involvement back a step into helping users annotate data? And we can see that in the next slide. And then after that, you can go again. Yeah, okay. So traditionally, images are annotated by humans. For example, a human whose job is to draw boxes around all the urchins in a large data set. What if AI can learn as the human annotates and use that information to help humans annotate faster? We are developing a way of having the AI work with the human annotator in real time, learning at the same time as the human annotates. This type of real-time collaboration has never been fully explored before. To help us study this setting, we developed simulators to observe how the system behaves without human involvement. Next. Okay, I think if the video is playing, okay. So here you can see the video of the interactive annotator. The purple boxes indicate boxes around urchins drawn by humans. The new addition to this interface are the red boxes. These red boxes are boxes that were predicted by the machine learning system. We have visual cues to indicate what the machine is thinking, like boxes will shake before it is deleted. As we can see, these boxes are initially not very accurate because it's early in its learning process. As the process continues, the machine should learn better and predict more accurately. Next. So the video you just watched showed a problem where we are detecting sea urchins. On the left, And we have two other problems that we are testing the annotator on. It will be detecting invasive Myconia trees and rapid ʻŌhiʻa death on Hawaii Island. This allows us to see how the system behaves in different environments. Next. So we measured the performance of the predicted boxes generated by the machine across all three data sets. Looking at the results, it would take a long time for the system to produce accurate predicted boxes, despite the large size of these data sets. In fact, the rapid ʻŌhiʻa death data set that we see that there were no useful predicted boxes that the system produced. We need the system to produce high quality boxes early on to provide useful help to the user. These results suggest that involving AI in annotation is not useful at all. Next. Due to the poor performance, we investigated what caused the system to perform so poorly. We found that the performance was related to the confidence of its predictions. For every box the machine produces, the machine also has a confidence. For example, the machine is 80% confident that the box it predicts is an urchin. The standard approach is that if the machine is above a confidence threshold of 50%, it will issue a prediction. However, when the machine begins learning, it does not have much data to learn off of. As a result, the machine confidence will almost never reach above that 50% threshold. So, also, how quickly the machine learns depends on the data set. So we created a system that automatically adjusts the threshold that makes use of the machine's predictions, even when it's not very confident. Next. Before we see the updated performance, let's just have a quick look again at the initial performance again. And in the next slide is the performance of the modified threshold, okay? We see that the performance improves across all data sets, and we see that performance improves really quickly for the urchin and Myconia data set. This means that the machine can quickly assist the annotator in creating boxes. With the modified threshold, we see that the raw data set on the right, top right, has significantly improved from its initial performance of 0. This means that at some degree, AI can assist the user in annotation. The graph indicates that it learns at different rates for each data set, which supports our idea of having the threshold automatically adjust based on how quickly the machine is learning. Next. So we just talked about incorporating AI machine learning into the annotation process and how we can make it faster. In order to annotate data, we will need to have the data first. Where does that data come from? Typically, gathering data involves a lot of time and human effort. Next. With the past project, we pushed AI into data annotation. In the next slide, we are going to push it back again into data collection. The next slide, sorry, and then you can go again, yeah. So there are a bunch of challenging domains for data collection. We chose to study data collection in marine science. For instance, divers collecting data on fish behavior are faced with a very challenging task. Next, please. One of the reasons it's so challenging is that there's so much information that divers have to be aware of underwater. We want AI or artificial intelligence to reduce the cognitive load of the diver. There are two challenging tasks that the divers face when dealing with data collection. Divers need to be able to detect fish and track them in a moving environment. This can be very hard to do when there's a lot of underwater life, which is why we want to explore how artificial intelligence can help ease this burden. First, there needs to be a platform to study how AI and humans can work together to collect this type of data more efficiently. Next. So to have a platform to study this, we need a way for machine learning to interact with divers as they are underwater. Our approach was to make a smartphone app that can be used in a dive housing. These requirements for this project are that it must be able to run on Android and desktop, track and detect multiple fish in real time on phones, and it also must be able to use any online Python trackers without modification. As of right now, there is nothing that fills these requirements. When investigating how to fill these requirements, we found that it was extremely challenging. And in the next slide, we can see a diagram of the fish tracker application. This implementation was not our first attempt, and it took us many attempts to design all the components needed to meet our requirements. And you can see that there are 5 different languages that they're communicating with. And then next, please. So here's a video of how the current fish tracker works. On the phone, we see that there's a box around the fish it found, and we can move the phone and the fish will be detected and tracked in near real time. You can also interact with the application by either selecting videos saved or using the camera. There isn't really a technology like this where it's actively tracking in real time on a mobile device. Next. So for the future, we presented new platforms that incorporate AI into data collection and annotation. This provides a gateway to studying new ways for humans and AI to work together on these challenging problems that have never been touched before. This just scratches the surface on the potential applications for human in the loop AI. Next. And I definitely couldn't have done this without my team members, Kian and Maynard. They have put in a lot of effort into this research and they have helped make this possible. I also want to thank Professor Mandel for this research opportunity, along with our collaborators, John Burns, Ryan Peroy, Bobby Suarez, and Tim Grabowski. This concludes my presentation and thank you for listening. Thank you. What are some of the difficulties for developing a UI that worked with underwater housing? Let's see. There's a lot of it's hard to get it working in the first place, I guess. So that was very challenging at first, and then let's see. We need to, and having the application work and on different devices other than a mobile device is also challenging as well. And I think that's all I know, because I primarily worked on the interactive annotator and Maynard worked on the fish tracker. Oh, sorry, I can't get more information than that. Yeah.
Yeah, if anybody has any questions, feel free to put it in the chat or put into the Q&A feature, and we'd be more than happy to relay it for you. We've got a couple minutes for questions, so if you have any questions for Ryan's, please don't be afraid to ask.
Oh, I think for John Graves, I think Travis is typing an answer to help answer your question. Yeah. Oh, why did Rod perform so poorly? I think it had something to do with like the colors or the consistency of the images. I think okay, I remember this. Okay, so the annotations I think generated by the humans were very inconsistent, and so that's why it performed very poorly when it tried to learn from the annotations from the humans. Yeah.
Okay, I'll open up the floor for any last minute questions. And if not, if you don't have any more questions, we'll move forward with our next presenter. Okay, sounds like that's it. Again, mahalo Ryan for that awesome presentation. Our next presenter is Zach Taylor with his presentation entitled Reassessment of Coral Reef Fisheries. And now I'll hand it off to you, Zach.
Thank you, ʻŌhiʻa. As she mentioned, yeah, I'm here to discuss reassessment of the methods, essentially, of coral reef fish surveys. Basically, the goal is to develop new methods that will be more inclusive of both the community as well as the fish communities as well on the reef. Next, yeah. So traditionally, and for the longest time, fish surveys have been conducted as fish, especially here in coastal communities, play a vital role for sustenance as well as just a livelihood. But fish, as we know, are not important just for people. They're also very important for the health of the reef as well. They play many key roles. They're predators and grazers on the reef as we out there in many different ways. They're also there to excrete nutrients. They're basically there to keep a balance overall. So for that reason, we've been monitoring them using two different types of gear over the years. Now there's active gear, meaning the gear goes towards the fish, such as a diver swimming along and counting. There's also passive gear where the fish is still or where the gear is still and the fish goes towards the gear. Now in a complex reef habitat, we're typically going to be going with the active gear. And traditionally we use scuba diving, and we do underwater visual transects where the diver swims along a transect and count all the fish within a certain area. And then we can do some calculations to do some biomass. However, this method does have some limitations we found over the years. And most importantly, what we have found is it can be invasive towards some fish and also biased in a way. When you're scuba diving, you're releasing a lot of bubbles, which can cause some fish to detour away and will not be ever included in your counts. It can also be expensive. As you see, it requires quite a bit of gear. And for that reason, also, it can be exclusive as it requires certifications also. So one proposed solution is using rebreathers, which results in no bubbles coming out as you're diving, which can definitely be less invasive. However, as you can see, it requires quite a bit more gear. So although there's no bubbles, there's a big trade-off because there's not a lot of people who have these certifications as well. And as you can see, can afford the setup also. So this method is great when you're doing deep reef surveys, especially, but in a shallow reef, it's not really a reasonable solution. Now, another proposed solution people have used is camera traps. Now, camera traps are used in many different survey methods. And for one, they are less invasive than a diver. They can be less expensive if they are maintained over time, and it does allow for an increase in data. However, traditional camera traps that have been used have some drawbacks also. One is they are typically not permanent structures. As you can see here, depicted by these arrows on the top, there's a line tied to it. That's to move the camera around and replace it. There's also a chum bag. Now this chum is going to result in an obvious bias of certain fish that will be attracted. So it's not giving you an accurate representation of the fish on the reef. So what do I propose as a solution? Working on two different methods. One is a free dive survey method, and one is using a live stream camera that is permanently mounted that will reduce that bias and that alteration in fish behavior. So first off, free diving. Why free diving? Well, free diving is much more accessible to many people, especially here in Hawaii, where there's a lot of spear fishermen who are out there surveying all the time. If we can develop methods, we can get a lot more data that way, and also be less invasive as we don't have all that gear in the bubbles to scare the fish. As you can see here in this image, the diver is much more blended in with the environment, which also will allow the fish to be more comfortable on these surveys. Now, the methods of doing free diving are going to be very similar to scuba in the fact that we will still lay a transect. We'll swim along that transect line, and every 5 meters we'll drop down. We'll hang there for about 15 to 20 seconds, count all the fish within that area, resurface, and repeat this all the way along the transect. Now, the hopes is that we can see similar results as these scuba surveys and can prove that this can be another viable alternative to scuba in getting more data. Now, the live stream camera, the big difference with this compared to the other camera traps that have been used is this is permanently mounted. So being permanently mounted, again, it's not going to alter that fish behavior. So we're getting a much more accurate representation of what the reef is like and also the fish that are out there. Because it's live stream also, we are going to get unprecedented amount of data, and also it results in a lot of inclusion. You can watch this on YouTube all day long. As long as the sun is out, you can see fish on the reef. So it really gives a new opportunity of surveying these reefs. In this image, you can see here, the bottom left arrow showing you typically over 100 people are watching this camera at a time. There's also a chat feature where they can be leaving notes and timestamps of interesting things going on, which really helped me out to see what went on that day on the camera. So this is going to be very useful, especially when we're doing these present pseudo-absent surveys. That's going to be the first thing we'll do. And we're going to compare these with 5 years of scuba surveys that have been conducted there previously to compare what fish are we missing. We have a hunch that we're missing things like the predators, such as the jacks, and also things like goatfish, which are typically flighty on a scuba survey. So the goal is to figure out which ones might we be missing and aren't included in these traditional fish counts. Now, this is also going to be useful because we can look at habitat preference more as we map this area. This also has potential, as Ryan mentioned, with AI as well for more data. So I'd like to thank my advisor, especially Dr. John Burns, for the work and for setting up this camera that has allowed this unique opportunity. And by using scuba and freediving and camera, the goal is to be able to integrate the most effective fishery management method. And with that being said, I'll open up the floor to any questions.
Mahalo, Zach. And yes, if you have any questions, again, don't be afraid to put them in the chat or in the Q&A feature, and we'll be happy to relay them for you. It seems like in the chat, John has a question. Is it possible to tell if you are double counting individuals?
So that is one of the things right now. We're not doing actual abundance counts. That's why we're starting with the presence absence more, because it is difficult. And as they're working on developing AI as well, that is something that has to be accounted for. Obviously, it's hard to tell, right, one yellow tang from another. So in terms of abundance, it's not quite there yet to necessarily say what the abundance is. More so, yeah, just what fish are there and what are we missing. I see Pat commented as well, increase the sample size to other reefs. Yeah, absolutely. that would be great. I'm sure we'd all love to expand that going forward. And then Dr. Proy, let's see, when do you think you have some results? So we're looking on some preliminary studies right now. Basically, we're looking at the saturation point of which we don't see any new species, which we found to be about an hour. After observing for an hour, you'll see about the max number of species you'll see. So we've noticed goatfish, you see a significant difference in, and the jacks, like I mentioned, are the two so far. But yeah, through the summer, we're going to be out there doing a lot more surveys as well to increase those samples. So hoping to have some early results next fall, and then yeah, presenting next spring again in the final results. And then how does this method account for when species are just absent from habitable area? So that is one thing we'll have to take into account. The one thing that we can compare though, because we have those scuba surveys there, we know what to compare to, I guess. So we can't necessarily say absent, more pseudo absence is what we'll be specifying, because as you mentioned, right, we can't, it's not the right habitat for every fish. But what we can say is we've been surveying it for 5 years, and just in the year of watching on a camera, we see this fish regularly all the time throughout the day. So we know it is there, but it is just not being accounted for on these traditional scuba surveys.
All right, if you have any more questions for Zach, please put them in the chat as we move forward with our next presenter. Again, mahalo, Zach.
Yeah, thank you, everybody.
Our next presenter is Carson Green with their presentation entitled Prediction of Macroalgal Species Composition Using Macroalgal Abundance, Case Study on Reefs in the Northwestern Hawaiian Islands. And I'll hand it to you, Carson.
Thank you so much for the introduction, Anohe. Yeah, I'm Carson Green, and today I'm going to be presenting on my senior thesis project dealing with macroalgae in the Northwestern Hawaiian Islands. Next slide. So what are macroalgae? Well, they're eukaryotes that are photosynthetic and multicellular. They're much larger than other single-celled algae you might know, like phytoplankton. And macroalgal distribution is influenced by a variety of factors such as things like temperature, water clarity, nutrients, and currents. Next slide. For my study, I am specifically looking at macroalgae in the Papahanaumokuakea Marine National Monument. The islands that are included in my study include French Frigate Shoals, Lisiansky Island, Laysan Island, Pearl and Hermes Atoll, Midway Atoll, and Curie Atoll. Next slide. There are as many as 500 species of macroalgae in the Northwestern Hawaiian Islands, with many recently being described. This happens as organizations have started conducting annual reef expeditions to remote reefs in the monument. And there are abundant algal functional groups, such as things like crustocoralline algae, turf algae, and fleshy macroalgae. But sadly, there are fewer studies on macroalgae in the Northwestern Hawaiian Islands compared to other benthic organisms, such as coral. So it's essential to fill in this gap of knowledge. Next slide. Here's an example of some of the common groups you might see in the Northwestern Hawaiian Islands. So in the top left, we're looking at filamentous turf algae. In the top middle, we're looking at chondria, which is not the white dead coral in the middle, but actually to the right of it. And then the top right, we have Halimeda species, and then we have some examples of CCA in the bottom. Next slide. So for my study, I was using data that was collected from 2017 to 2019 on cruises. And on these cruises, teams of divers would go down and they would lay down transect lines about 0 to 30 meters deep on the reefs. And they would collect overlapping imagery along each of these transect lines, aiming for about 70 to 80% overlap between images. Next slide, please. So with this overlap, we were able to use these images to construct three-dimensional models of survey sites. And this was done using Agisoft PhotoScan. So with these images, we created a dense point cloud. And from those images, we then stitched it together to form these orthophoto mosaics, which you can see at the end of this process on the slide. Next slide. Using these two-dimensional orthophoto mosaics that were stitched together from the survey sites, we then uploaded them to CoralNet, where, next slide. Where for each plot, 1,000 random points were generated for annotation. So for each point, all benthic features located were labeled, and all features that could be labeled down were classified down to genus level manually. From this, percent cover was calculated for each survey by dividing the number of points for each category by the total annotation points. Next slide. We also looked at the data using ArcGIS to check ideas if it was too hard to see on CoralNet, and also to look at spatial reference. Next slide. Here is a chart looking at some of the main groupings found in my study. In colors, we see the three main databases where I got data from, which includes Ramp, Benthic, and Pearl and Hermes, which include all 6 islands. One thing of note on this chart is looking at turf algae, we can see high percent cover values for all of the databases. Another thing of note is looking at Rhodophyta red algae, we see a large percent cover compared to the others for Pearl and Hermes, which can be associated with more recent chondria outbreak in Pearl and Hermes atoll. Next slide. Here's a look at percent cover for all of the labels in my data set. So looking at the top left is a pie chart for all islands in my study. In bright blue, we see that turf growing on hard substrate was the macroalgae with the highest percent cover, followed by CCA growing on hard substrate in the pinkish color. Then we had turf growing on rubble and CCA growing on rubble as well. But something that of note for this is looking at Lisiansky Island in the bottom left, we can see high value of Halimeda. And in Pearl and Hermes, in the bottom right, we see high value for chondria species of algae, which kind of explains some of the high values when we were looking at all macroalgae for the top left pie chart, because we have such high values in both Lisiansky and Pearl and Hermes for those macroalgae. Next slide, please. Here's a graph looking at percent covers between all 6 islands in my study. For French frigate shoals, we have the most variability and percent cover of all the islands, but also the lowest median value for percent cover. Relatively, Curie Atoll has the highest average percent cover of all the islands. This is interesting to note because of the latitudinal positions of French frigate shoals and Curie Atoll, with French frigate shoals being the most south and Curie Atoll being the most north. Next slide. Here we see a strong negative relationship between algal percent cover and coral percent cover in all the islands. Here, high values of algae is associated with lower values of coral. This is also been studied before and been looked at when comparing competition for benthic substrate between coral and algae. And it was nice to see it in this study. Next slide. Here is another regression model looking at cressus coralline algae, CCA, and turf algae. This, we only have a moderate negative relationship, and where higher values of CCA are associated with lower values of turf algae on the islands. Next slide. When looking at these relationships, I also wanted to see how they changed between islands. So I made mixed effects models. Here, something important to note is to look at islands such as Midway Atoll when comparing coral and algae percent cover, which actually does not show a strong negative relationship like all of the other islands in the study. Next slide. Next slide, please. Oh, thank you. Sorry. Oh, wait, yeah, go back. Thank you. Here's another mixed effects model of the regression we saw earlier between CCA and turf algae. Again, not all islands show this negative relationship between CCA and turf, where islands like in red French Frigate Shoals and in the darker blue color Midway Atoll showing almost no relationship between CCA and turf percent cover. Next slide. So for conclusions, turf algae and CCA are the dominant macroalgae in the Northwestern Hawaiian Islands, and the total algal percent cover varies significantly by island. There's also a significant negative relationship between algal cover and coral cover, and a significant negative relationship between CCA cover and turf algae cover. In the future, I was hoping to examine how the macroalgal community composition related to the overall macroalgae cover on these islands. Next slide. Thank you to my advisors, Carla McDermott and John Burns, also Kylie Pascal and Alex Spangler for their help on this project, and all other Mega Lab members who helped me annotate the coral net plots for this study. Next slide. Mahalo, and I'll take any questions. Thank you.
Mahalo, Carson, for that awesome presentation. And yeah, like he said, the chat is open for any questions. Seems like we've got a question from John. And his question is there a consensus on why Midway and a few others had no correlation between coral cover and algal cover? Is the benthos just significantly different at these atolls?
Yeah, that's a really interesting question, John. When reading it, I don't think I saw anything about or like a consensus on why they had no correlation on those islands, but it's definitely something that would be really interesting to look at, especially for a study like this. There's a lot of factors that go into why the algal cover might be different among these islands, or why there's a different relationship between the algae and the coral. So it's definitely something I would like to look more into, but I don't have like a definite answer on why there's no correlation between it on those islands right now, but hopefully we can figure it out soon. Oh, no problem, John. Thank you.
Seems like we've got at least 1 more minute for questions if anybody has any.
Ooh, what was I most surprised by in your results? I think going into it, looking at the relationships, there's also a couple other relationships I didn't get to share today because of time and stuff, but I think the biggest was looking at some of the regression models and the relationships I saw. I hadn't seen too much about, like there's some things that are, you might expect to see, like the coral and algae relationship, but things like CCA and turf were not actually relationships I was expecting to see going in. And there's a couple other relationships I was pretty stunned by when looking at this. So just after doing the stats and actually seeing those significant relationships on the islands, I think probably CCA and turf was one of the most surprising for me, just because it's not something that's as talked about in the literature. So.
Okay, I think it's time for us to move forward. Again, mahalo Carson for that awesome presentation. And feel free, if you have any more questions, just put them in the chat and I'm sure he can get to them later. Our next presenter is Olivia Jarvis with her presentation entitled Observing Patterns in Hilo MOP Turtle Tagging Data Through Statistical Analysis. And now I'll hand it off to you, Olivia.
Thanks, Anu. Aloha, everyone. My name is Olivia Jarvis, and for my mock project this semester, I worked with the Hilo mock turtle tagging dataset to find patterns within these records collected over the past 40 years. Any analysis of this long-term dataset had yet to be completed, so I took on the task for this project. Next slide. NOAA sea turtle biologist George Balaz was interested in studying Hawaiian green sea turtle populations and worked with HPA and Hilo MOP to tag and assess green sea turtles at Punaluu Beach Park to track growth and monitor population size. Next slide. The goal of this project was to take this large data set, organize it in a way to conduct statistical analyses and look for trends among what had been collected so far and identify directions for future data collection and analysis. Next slide. Some of the important information collected at these tagging events included the pit tag ID, sex, presence of tumors, along with measurements of straight carapace length, curved carapace length, and weight. These three measurements are the key indicators of size for turtles in this data set. Next slide. Most of the data were entered into an Excel spreadsheet that I imported into RStudio, and I used functions designed to fix typos, remove and add any columns, and filter out any missing values. Next slide. And for the statistical analysis, I conducted summary statistics and I grouped turtles by sex and tumor presence to make comparisons. And I also tested for correlations between any of the measurements. Next slide. So from this, I was able to gather that turtle tagging records began January 20th, 1978 and are up to date through July 9th, 2018. A total of 1,220 observations were recorded, and of those, 821 observations had measurements. About 434 different turtles were tagged, and over the 40 years, 99 different dates of observations were recorded, with observations ranging from 1 to 23 turtles recorded on a given day. 23 of the turtles were found with tumors, and 790 did not show any tumors. And of the 821 turtle observations with measurements, 5 were identified as females, 26 were male, and the remaining 790 turtle sex were unidentified. Next slide. These were the overall distributions of SCL, CCL, and mass. The blue line shows the mean, the red line shows the median. So for straight carapace length, measurements ranged from about 20 to 90 centimeters with an average of 61. Curved carapace length measurements ranged from 21 to 95 centimeters with an average of 65. And mass measurements ranged from 2 to 115 kilograms with an average of 33. Next slide. And here are the distributions of SCL, CCL, and mass for males in blue versus females in pink. Visually comparing the groups, there are very minor differences, but we see females being slightly larger than males on average, but the Wilcoxon signed rank test shows that there's no statistically significant differences. Next slide. And similar results are seen when comparing size among tumor presence in turtles. Visually, those without tumors in green are slightly larger than turtles with tumors in orange on average. But the Wilcoxon sign ring test shows there's no statistically significant differences. Next slide. And when plotting each size characteristic against the other, the Pearson correlation test demonstrated there is a relationship between SCL, CCL, and mass, and there's strong positive associations. Next slide. And overall, the results of this analysis provided some insight into the long-term study of Hawaiian green sea turtles at Punaluu Beach Park. However, something important to note with these comparisons is the sample size. Although 821 observations of carapace length and weight were recorded, majority of the records were unable to visually identify sex of turtles due to them being sexually immature, which left 5 females and 26 males as samples to represent the entire population of Hawaiian green sea turtles at Punaluu. So confident comparisons between average male and female measurements can't quite be made from these data. But a possible way to ID a few more males and females could be if turtles have been caught more than once over the years, recorded as juveniles as first. We may be able to go back and record male or female now that they've been recaptured and possibly sexually matured. Next slide. And overall, it's important to continue expanding this data set to better understand growth rates and gain insight into environmental conditions Hawaiian green sea turtles are faced with. And having tag records can help wildlife managers and researchers keep track of nesting females and population size. Next slide. Thank you to UH-Hilo and all my advisors, and thank you to TCBES for hosting the symposium. And I'll take any questions. Let's see if curve carapace length, straight length are correlated. Are they both measured? And one tell you. Straight carapace length kind of gives the overall size of the shell, and curved, there could be a larger hump or it's slightly larger than straight carapace length. They did measure a lot of variables in this data set, like tail length, the front side of the shell. But yeah, so they measured a bunch of things. So this was just one that was entered into the data set, but I don't think there's any major differences. Do you think these turtles are residents of Punaluu or moved around a lot? How does these? So tumor levels actually in Punaluu are lower compared to the other islands. I think they see more in Oahu from what I've heard. And I think most of the turtles at Punaluu are resident, but they do travel around and they're doing more tagging events or just like observation events on other islands. They have, I read a paper, I think there's 6 other sites in the Hawaiian Islands that they observe turtles at. And so I think we're working on building a photo ID data set to maybe see if like these turtles are just at Punaluu or if they do move around and found at other sites. So that could be interesting too, to see if are these turtles really residents. And then do you think there's any opportunity to use this data set for any kind of machine learning? Yeah, I think if the photo ID data set, one of the other MOP students is starting to work on this. That could be really interesting. We're learning deep learning with images and machine learning now. But yeah, there's still a lot of different things that can be done with this data set that, yeah, statistically or machine learning wise can definitely look at, yeah.
Well, it seems like we're running short on time, but mahalo, Olivia, for that awesome presentation. And our next presenter is Walter Boger with their presentation entitled Shifts in Carbon Exports from a Hawaiian Watershed Under a Changing Climate. And I'll hand it off to you, Walter.
Thank you, Anu. Yeah, so my name is Walter Boger, and I did a study on shifts in carbon exports from a Hawaiian watershed due to climate change. My advisors were Dr. Colburn and Dr. Weaker. Next slide. Yeah, so rivers are important pathways for carbon into the ocean. And there's several forms of carbon species that are transported this way, and that's dissolved organic carbon, dissolved inorganic carbon, and particulate carbon. And it's important to understand the rates and the sizes of these carbon inputs for two reasons. One, it's important for carbon budgets, and two, it's also important for ecological reasons. And so if we know that these carbon sources are large, we can regulate them. And if they're sinks, we can enhance them to kind of solve partly our carbon problem. And the other ecological reason to understand why these carbon importance are kind of quantitatively study these carbon exports is that food webs use this organic matter as a source of food. Next slide. And so carbon transports are kind of changing due to climate change. The rainfall distribution over a time scale is changing. Where certain parts of the year are receiving more rainfall or less rainfall. And this kind of translates into a watershed change as well, where there's durations of droughts are increasing, or the droughts intensity is increasing, or the frequency of storm flow is increasing, and the intensity of that storm flow. So there's overall changes in river discharge. Next slide. And so specifically here in Hawaii, there is some predicted changes for the rainfall pattern, and that's the increase in the duration of droughts and increase in the severity and frequency of storms. So our storms are getting more severe and they're more frequent, and the period between those two storms is becoming drier. There's also another rainfall predicted change, and that is that the elevation of the rainfall is decreasing. And so these changes could disturb coastal marine food webs as rainfall kind of transports all this material into Hilo Bay, for example. Next slide. And so that kind of arises to our question: how does river discharge influence delivery of inorganic and organic carbon in origins to Hawaiian estuaries? Next question. Next slide. And so to answer that question, we looked at the Wailuku watershed. And so the Wailuku watershed is the largest watershed in the state of Hawaii. It's 576 kilometers squared, and it drains parts of the world's massive mountains, Mauna Kea and Mauna Loa, into Hilo Bay. The Wailuku River drainage area is about 76% native forest reserve, 20% pasture, and 4% urban area. It's located on the eastward side. As you can see it here in the bottom figure, that it drains Mauna Kea and Mauna Loa into Hilo Bay. Next slide. And so Wailuku in Hawaiian language, Wai means water and Luku means destruction. And so the river is the waters of destruction, and it's a pretty fitting name. Some basic information about Wailuku river flow is that base flow and storm flow, the cutoff is 3,100 liters per second, but max flow has been 2.3 million liters per second. And so there's a great bit of variability in how intense this storm or flow can go. Here on the right side, we can see a hydrograph from September of last year to, I think, November of last year as well. And on the vertical axis, we have discharge per liters per second. And on the horizontal axis, we have date. We can see that these peaks, when it rains, it skyrockets in discharge, and it's pretty quick. It goes up and down very fast. And on these stars, that's where we sample. And our goal of sampling is not necessarily to capture a single storm event, but to just get a range of discharges, trying to get on the left or the rising or the falling limb of the hydrograph. Next slide, please. And so our experimental design is to get 25 water samples over a range of flows. Currently, we have 740 to 3,200 liters per second, and to examine the relationship between carbon species and discharge. And so we're measuring particulate carbon, total alkalinity, which you're using as a proxy for dissolved inorganic carbon, dissolved organic carbon, and we're also looking at carbon-13 and DIC, PC, and DOC. And we're also looking at oxygen-18 and deuterium in water as a proxy for rainfall elevation. And we're using carbon-13 stable isotope and DIC, PC, and DOC to eventually do a stable mixing model to find out the source of the carbon. So we're also looking at physiochemical water properties as well of turbidity, specific conductance, temperature, and oxygen concentration. Next slide. So simultaneously, we're also doing 25 drift net samples over the same range of flows to examine the relationship between coarse particulate organic matter, which I'll call CPOM, and flow as well. So we have a drift net that's 250 microns, and it's equipped with a mechanical flow meter. And we collect debris. Currently, we've collected wood, barks and twigs, leaves, ʻŌhiʻa, mostly Albizia, seeds as well, terrestrial animals, we have chicken feather, some fish as well, and filamentous algae and sediment. And next slide. So here's some preliminary results. So this is total alkalinity, which decreases as a power function with river discharge. And this is a similar relationship that was found in tropical rivers in Australia. And again, we're using this as a proxy for DIC. In the future, we're going to look at bicarbonate and carbonic. And so, yeah, DIC decreased with river discharge. Next slide. And then we had a DOC concentration increase with river discharge in a linear relationship. And this is probably due that during storm flow, water shifts to shallow flow paths as the soil column gets saturated, and it flushes all that litter and upper soil into the water. And it's also maybe due to the fact that the scouring of the stream bed is kicking up a lot of material. And so here on the figure we have on the vertical axis, it's DOC concentration per micromoles per liter. And on the horizontal axis, we have liters per second in discharge. And we only have 8 samples currently. We're waiting back for more from the analytical lab. And using this linear relationship, we can actually predict DOC concentrations as a function of discharge. And fortunately, the Wailuka River is equipped with a USGS water watch gauge. And so it takes discharge readings every 15 minutes. And so using that data, we can actually calculate DOC concentrations and then DOC concentrations times the discharge. And we can integrate that over a time span and we can get carbon fluxes or DOC fluxes. And so we did that. And next slide. We did that from August of last year to February of this year. And we found that DOC influxes are entirely influenced by storm discharge. And so on the left figure, it's actually on the vertical axis is log. So there's a great amount of variability of DOC exports. In October, we found that there was about 400,000 tons per that month. And in February of this year, it was very dry. We only had 22.5 tons per that month. And on the right-hand side, using the cutoffs from previous 3,000 liters per second, we can contribute what amount of that DOC flux is due to storm or base flow. And we found that in February, there was no storms. And in October, there was a lot of storms. Of 99.97% of the time, it was storm flow. And so with a changing climate, we could see maybe DOC exports are expected to increase if the intensity of these storms are expected to increase. Next slide, please. And so we also looked at coarse particulate organic matter, and this is mass, and it changed with river discharge. We have base flow on the left-hand side and storm flow on the right-hand side. The main difference between the two is that in the base flow, we found aquatic animals and terrestrial animals. We only found 3 fish, and so it probably offset our data by a lot. And so the terrestrial life as well, if similar, we only found the chicken feather, not a chicken leaf. And on the storm flow and the base flow, there was kind of a relatively equal distribution of leaves, wood, seeds, and sediment. Next slide, please. And so our conclusions is that storm flow has a higher and different composition of carbon than base flow concentrations. We saw that linear increase in DOC concentration, saw the power function in the total alkalinity, and we found that there was different terrestrial and aquatic life in the CPOM data. And because of that, a change in climate could change the amount and composition of DIC, DOC, and CPOM. As weather is, rainfall patterns are expected to increase in severity and frequency, and the duration between the two storms is supposed to be drier, there's going to be changes in our exports of carbon. And so what our future work is to continue to collect data to refine these models. We still have a lot of data to collect, and it would be better to have those. And then make predictive models for carbon fluxes based on climate change predictions. So we're going to use the literature that's out there that's expected to change the rainfall patterns and use that in our model to now calculate carbon fluxes. Next slide, please. And so I'd like to make acknowledgements to all my funding agencies and all the people who have helped me, and I'll take any questions now.
Thank you, Walter. Actually, I do have a question for you. I'm just kind of curious. What are some of the consequences of increased carbon exports in our watersheds?
So the consequences of increased carbon exports. So a lot of this carbon is particulate carbon. It could be, maybe increased turbidity in the water column, which might affect photosynthesis. It could be, so there was a paper by Atwood, which found that during storms, you have this carbon export a lot of it goes to the primary consumers and not the secondary consumers. So there could be consequences in the food webs, where these food webs are necessarily impacted by how storm changes. Yeah, so there's some negative consequences that could be there.
Thank you. It seems like we also have a question in the chat, Scott. And they say, if DOC exports increase with expected future increased storm intensity, do you feel drying intensity between storms will be linked to carbon export in watersheds? And is there existing evidence for this?
Let me read the question, sorry.
No worries.
Sorry, I'm just reading the question right now. Storms will be linked to carbon. Sorry, I'm maybe having a little trouble understanding the question. DOC exports increase with expected future increased storm intensity. Do you feel drying intensity between storms will be linked to carbon exports in the future? Can you rephrase the question a little bit? I'm sorry, I'm having a little trouble understanding.
They might be asking if, or, oh, we have clarification in the chat.
Oh yeah, sorry. Oh yeah, that's a great question. Yeah, yeah, probably as we're probably buildup of a lot of more of this organic matter, it probably will be flushed and increase the amount of carbon. Yeah, I don't know if there's any understanding literature about it, but it's definitely a good question. That's something I should definitely look into. Yeah.
And if we don't have any more questions, I think it's a good idea to move along into the next presentation. Again, thank you, Walter, for that great presentation. And our next presenter is Sofia Smith with their presentation entitled "The Distribution and Abundance of ʻŌhiʻa-Associated Ambrosia Beetles in 2 Sites with Rapid ʻŌhiʻa Death Outbreaks." And I'll hand it off to you, Sofia.
Thank you for the introduction. Hello, everybody. My name is Sofia Smith, and I'll be presenting my project on the distribution and abundance of ʻŌhiʻa-associated ambrosia beetles in 2 sites with rapid ʻŌhiʻa death outbreaks here in Hawaii. You can go to the next slide. So to start us off, I'll introduce you to the key species involved in this project. The first one I'm sure we're all familiar with is the ʻŌhiʻa lehua. They are endemic to Hawaii, meaning they're found nowhere else in the world, and they comprise an estimated 80% of remaining native Hawaiian forests and provide critical habitat to other native and endemic species, including the ʻiʻiwi, which you can see in the top photo right there. So conservation of ʻŌhiʻa is critical to conservation of Hawaii's native forests as a whole. You can go to the next slide. Unfortunately, ʻŌhiʻa have recently become threatened by an introduced disease that kills ʻŌhiʻa called rapid ʻŌhiʻa death. It is caused by 2 different species of the fungus Ceratocystis, which are Ceratocystis leuciohea and Ceratocystis huiohea. The disease spreads by entering any open wound on the ʻŌhiʻa, and the fungus starts to colonize the xylem of the tree, which is what the tree uses to distribute water and nutrients throughout it. And rot is easily identified by the reddish brown color the leaves start to turn before they fall off, which you can see in the photo on the right. And once a tree starts to show symptoms, there are no treatments available, so it's already on its way out. It was first discovered in 2014 on Hawaii Island, which is where I'm from and where the study takes place. And rot has also unfortunately spread to other islands and was discovered on Kauai in 2018 and Oahu and Maui in 2019. So we assume rot is spreading inter-island because of people transporting ʻŌhiʻa wood between the islands, which we've managed by requiring a permit to transport ʻŌhiʻa wood. But understanding the spread of rot on a smaller scale is critical to mitigating any further spread, which brings us to the last species involved in my project. So you can go to the next slide, which are the ambrosia beetles. So they are a type of beetle that bore into the wood of trees and introduce species of a symbiotic fungus, which is different from Ceratocystis. And they usually go for dead or dying trees. And as they bore into trees, they produce a sawdust-like substance called frass, which you can see in the bottom photo. And ambrosia beetles are known to attack trees that are actively dying of rot, and they are known to spread plant diseases in other environments. For example, they've been linked to thousand cankers disease in black walnut trees in the eastern US. And for our study, we decided to focus on the species Xylobernius saxonii because it is known to use ʻŌhiʻa trees as a host, and saxonii are not native to Hawaii. You can go to the next slide. So because ambrosia beetles attack dying trees and spread fungal spores, it's been theorized that they could be another agent in the spread abroad by either carrying Stratocystis spores from an infected tree on their body to a new healthy tree, or by introducing viable spores into the environment via frass production, which could be carried by the wind or other animals into trees with open wounds. So a previous study showed that in a lab environment, Saxinii produce frass containing viable Stratocystis spores 64% of the time, and beetles that were exposed to a sample of Stratocystis can pick up those spores and then infect a healthy tree with those Stratocystis spores, which that experiment is what's going on in that photo right there. However, the pattern and abundance of these beetles and the rate at which the beetles carry rod in the wild are unknown. You can go to the next slide. So since we know that ambrosia beetles can spread rod in the lab, we wanted to know how they could be related to the spread of rod in the wild. And we decided to explore this with two questions. Are beetles more abundant in areas with rod positive trees? And at what rate are beetles carrying viable Stratocystis on their bodies? You can go to the next slide. So the study sites we chose to explore these sections were two sites within Hawaii Volcanoes National Park that were experiencing a rod outbreak, the Ola'a and Kahuku units of the park. You can see the location on the little map right there. And we established aerial beetle traps in a 75 by 75 meter grid in both sites. What the trap looks like is shown in the top photo. And they are designed to attract beetles using a lure of ethanol because it's chemically similar to what trees give off when they're under stress, and which is what attracts ambrosia beetles to them. And we also cycled the traps using water as a control lure. You can go to the next slide. So our first research question is, are beetles more abundant in areas with rod positive trees? So we wanted to know if the traps that were catching more beetles were closer to trees with rod. And to answer this, we have the data from our traps and the tramp samples that was collected weekly, and the beetles were counted by trap location and identified to the species level. But for the location of the rod trees, we use aerial survey data from the Hilo Spatial Data Analysis and Visualization Lab, which had the location of 23 trees, which were identified as either high or medium confidence for having rod. And of those 23 trees, 11 were sampled by the Park Service and all came back positive for rod. So for our study, we assumed that all of the identified trees were positive. You can go to the next slide. So this is a map of those trees in our grid of beetle traps in our Ola'a study sites. The red dots are high confidence trees for rod, and the orange are medium confidence, and anyone with a check mark is one that was sampled and tested positive for rod. So now that we had the number of beetles caught per trap and the location of the rod positive trees, we calculated the distance to the nearest rod tree for each of our traps and related it to the average number of beetles caught per trap. You can go to the next slide. So to visualize that data, we made this figure, which shows the location of the rod trees as the plus signs now, and the location of the traps as these black dots. And the bigger the size of the dot, the higher the average number of beetles captured for that particular trap. So as you can see, it doesn't really look like there's a strong relationship between a nearby rod tree and a higher beetle count. We expected bigger average beetle captures and bigger dots from those traps that are nearby rod trees, but that isn't really happening. You can go to the next slide. We also did a linear regression model on the distance to the nearest rod trees and the mean number of beetles captured, and this also verified a lack of relationship between the two. You can go to the next slide. So Kahuku's data also told the same story of a lack of relationship between the beetles captured and the distance to the nearest rod tree. The black dots did not get any larger the closer they got to these trees, and the regression again showed no relationship. You can go to the next slide. Since we didn't find any clear relationship between the location of the rod trees and the mean beetle captured, we decided to look at temporal variation in the number of beetles captured per week to see if there might be some seasonality involved in the beetle activity. So we did an autocorrelation function to compare one week's numbers of beetles captured to the previous week's number of beetles captured. And again, there was no relationship there. So it seems pretty random over time as well as over space. You can go to the next slide. So to answer our second question, at what rate are beetles carrying viable Stratocystis on their bodies? Besides just counting and identifying the beetles, we also took the entire sample, including the water and any debris that might have fallen in, and cultured it for Stratocystis, using carrots as a growth median for the Stratocystis, which you can see in the top photo right there. So those are what the samples look like when they are aerated. And if there is growth after 2 weeks of aeration, we check it under the microscope. And if it looks like Stratocystis, we confirm it with PCR DNA analysis. And the picture on the bottom is what we were looking for. So that's what rod looks like growing on a carrot under the microscope. And something to note about this method is that the sample collection tubes were not just exposed to the beetles, they were also exposed to the environment. So frass spitting in the wind could have also affected the rate of positivity we got. You can go to the next slide. So this figure shows the result of the viability analysis in ʻŌhiʻa. It also shows the location of the rod trees as the red pluses, but the dots are now traps that never had a positive viability result, and the triangles are traps that did have a positive. So only 2 traps in ʻŌhiʻa ever had a sample that was positive. The one up top only had 1 positive, and the one on the bottom had 3 positives over the course of our study. And what's interesting is these traps are really close to a rod tree. They have one in really close proximity. They have plus signs basically right on top of them. You can go to the next slide. Kahuku, again, had similar results, only 3 traps with viable samples, and they were only positive 1 time. However, the viable samples were not very close to any of the known rod trees, and the traps that are next to a whole cluster of rod trees never turned out viable. So again, there's a lack of relationship there. You can go to the next slide. So the information that we got from this data is that there's no relationship between the location of the rod trees and the amount of beetles captured. The beetle counts are highly variable, both temporally and spatially. And we also found very low viability results in the beetle samples we collected, but that viability does occur. So there is some sort of Stratocystis transmission happening, maybe on the beetles themselves, or maybe in the wind spread by their frass. We're not sure. You can go to the next slide. Some future research that could help clear up some of the questions include more research into the other species of ambrosia beetles that use ʻŌhiʻa as a host. So there's a native ambrosia beetle, Xyloborus similimus. However, we never really caught that species. We only caught 1 in all of the weeks and all the traps of our study. And maybe that's because the lure we were using isn't very effective on them. So looking into Similimus and how to capture them and look at their relationship with rod trees would be valuable, since they only use ʻŌhiʻa as a host, so they could have a much higher rate of carrying Stratocystis. We also don't understand the drivers of beetle abundance, since they are so varied over time and space. So looking into factors that influence that could also be really useful. We also don't know if the samples that came back viable and positive for Stratocystis were that way because of the beetles or because of frass in the environment. So determining the difference between those 2 methods of spread would be really valuable. And while the viability rates are low, if there's thousands of beetles, then that could still have an impact on the spread of rod. So looking at the efficacy of methods aimed to reduce beetle numbers or spread, like the felling of rod trees and tarping of the fallen trees, or beetle repellents, would also be a really good piece in the puzzle of figuring out the role of ambrosia beetles in the spread of rod and how to manage that. You can go to the next slide. I would like to thank my mentor, Helen Sofair, as well as all of my beetle team members, Robert Peck, Ellen Dunkel, and Jordan Sarders, as well as my collaborators from the SDAV lab, Timo, Ryan, and Nadia, as well as everyone on the PIPES team for all their help over the summer and continuing into the 2 semesters that I've been doing this project, Mina, Devin, and Sharon. So thank you guys so much, and thanks everyone for listening.
Mahalo, Sofia. And yeah, we'll open up the chat for any questions that you may have. I seen earlier that Katie had a question. She asked, or they asked, why carrots?
So carrots have been identified as just a really good medium for Stratocystis fungus in general, not just the species that cause rod. And they just really like carrots for some reason. So that's what we used as our growth medium.
And then John also had a question too. He says it would have to be a longer term study, but have you considered looking at the reverse relationship? Look at where you're seeing larger numbers of beetles and if the trees in those areas experience higher rates of rod over time.
That's a really interesting question. I could see that being a really valuable study method to use. The problem with looking at the rod over time is that it's intensive to take a survey of all the rod trees. A ground survey is really difficult in testing them, or the data we used from the SDAV lab, they were doing it with helicopters or drone footage, which is expensive, and they can't really just go out every month and do that. So it's hard to track rod trees over time, if that makes sense. So it'd be kind of hard to see how much they increase over time unless there was a really intensive study of aerial surveying of one area consistently over years. But I think it'd be a really interesting thing to look at.
Pat also has a question in the chat. He said, really impressive presentation. Sounds like these beetles aren't major vectors. Were they considered to be more of a problem in earlier years?
Yeah, so thank you. They definitely were a concern when rod was first being investigated, because we knew that ambrosia beetles were going for rod trees. And the study that I mentioned in the lab, it looked like they could be really spreading it. But yeah, maybe they were more of a problem in earlier years, or maybe we just weren't looking at the like wild transmission rates. But yeah, it looks like from the study, they aren't super big concern, but this is also very preliminary data. And I think it'd be hard to say that without a lot more studies in different areas, looking at all the ambrosia beetles and how they could be transmitting the fungus.
I do have a last very short question that I'm kind of curious about. Why did you guys pick Ola'a and Kahuku as your sites?
So those were chosen because they were experiencing a rod outbreak already, and they were in the national park. So the team that I was working on like had access to that and were permitted to go put up beetle traps. And they were also fenced areas that had already been documented by the SDAV lab, or Ola'a at least was already documented by the SDAV lab with the rod trees. And we knew from preliminary studies that they did have the beetles there and there was rod. So that's why we chose them.
Thank you. And I think that's all the questions we have. Again, mahalo, Sofia. And our next presenter is going to be David Russel with their presentation entitled Creating a Climate Adaptation Plan to Benefit a Community-Based Subsistence Forestry Area. And I'll hand it off to you, David.
Right on, mahalo. Yeah, aloha everybody. So yeah, I'm just going to be sharing with you all some of my work that basically just involving the creation of a climate adaptation plan that will benefit a community-based subsistence forest area.
Oh, sorry, David, I think you're muted.
Oh, I'm so sorry. I've been doing, I'd like to start by kind of sharing the mo'okuahau, or the genealogy of my study area. And it goes, o hualalai ke kupuna hine hanau ia o pu'uva'ava'a kamakua. O pu'uva'ava'a kamakua hanau ia o pu'uanahulu ke keiki. Next slide. So yeah, that basically tells us that Hualalai, the grandmother, first gave birth to Pu'uanahulu, that gave birth to Pu'uva'ava'a, the mother, and then Pu'uva'ava'a, the mother, gave birth to Pu'uanahulu, the child. And in geological terms, this is telling us, of course, that Hualalai erupted, forming Pu'uva'ava'a, and then Pu'uva'ava'a erupted, forming Pu'uanahulu. Next slide. So, as you can see on this map, Pu'uva'ava'a and Pu'uanahulu are actually also two ahupua'a in the kalana of Kekaha Vaiole o Nakona. And this is the kalana is a land division in Kona, and that the name Kekaha Vaiole basically means the waterless lands of Kona. And for obvious reasons, there's a lot of dryland forest here. Next slide. So Pu'uva'ava'akon, it's actually the oldest geological feature on the Hualalai volcano. It has a trachyte pumice composition, which is unique. It stands almost 4,000 feet above sea level, and on the surrounding landscape, it's over 1,000 feet tall. It also covers 460 acres, which is about a mile in diameter. So it's quite a large feature, and it's really hard to miss. I'm sure a lot of you have seen this in North Kona. Next slide, please. So this map is just kind of showing the area view of the Pu'u, and you can see in the red outline there is the Pu'uva'ava'a Community-Based Subsistence Forestry Area, or the PCBES, which is an 84-acre unit on the northeastern side of the Pu'u. That was really kind of conceived in 2017 when land managers from DOFA and USFS discussed with the lineal descendants of Kekaha Vaiole about creating this unit, this community-based subsistence forest area. So that unit was actually completed in 2020, and my project is going to help inform that. Next slide. So yeah, we have kind of 4 main goals for my project, which is first is to identify the historic plants, disturbances, and any climate data on Pu'uwa'awa'a. The second major goal is to just survey the current native plants, and by native plants, I really mean the remnant native species which have been there. And the third main goal is to really take this information from the surveys and the historical information and plan for future climate conditions by looking at what species are resilient. And the fourth main goal is that you're really taking everything from this process and finally creating a climate adaptation plan for Pu'uwa'awa'a that can be used for land managers and of course the community to just help inform restoration for a resilient future. Next slide, please. So yeah, when I started with this project, I really just started with gathering these various pieces of information, and there's a variety of resources we used, which include biological assessments from Pu'uwa'awa'a, books, cultural assessments, newspapers, and even the things like botanical survey notes. Also, the community meetings were a huge aspect, and we got the community and the lineal descendants input, as well as what they remember and what they know and what they want to see as well. Next slide, please. We also were able to find some old maps and some old images, as well as some rainfall data and that sort of a thing. Next slide, please. So yeah, we kind of, concerning the plants, I ended up creating this plant list with the help of another project member. But basically the plant list is just all of the plant mentions like that we can find in all of these sources that was found in each source. And of course we tallied the mention of each source and recorded the species, and then any other information along with that, such as like where that plant might have been found. And yeah, Pu'uwa'awa'a, the fact that we're doing this specifically on the Pu'u kind of made it a little bit tricky, because the Ahupa'a carries the same name. So, but basically I got all of the plants associated with the name Pu'uwa'awa'a to start with, and from there I got some more discrete information. Next slide, please. So this is just some images I wanted to share with you all that kind of show some of the land cover changes over the last 100 or so years. It's pretty amazing. But yeah, you can see the first image in 1900, just how different it looked from today in 2020. Next slide, please. And here we have some more images, a little bit more recent, but again, you can see there's quite drastic changes in just these few years, about what is this, 40 years or so. So yeah, I just wanted to kind of show you guys that this Pu'u has been changing, and there's a lot still to come. Next, please. So yeah, one of the big aspects that we're currently still finishing up is the ground surveys of all of these remnant species. So I'd say we're around 80% or so done. I'm gonna finish it up this summer and then get into some of the other stuff. Basically, that has just been entailing, taking these GPS points of these species and then getting the measurements, which include the diameter at breast height and the canopy spread, and then also recording any other relevant information, like if that tree is in a gully, or kind of if it's on one side of a ridge or the other side, and we're hoping to use that information for the future, like climate adaptation aspect of it. Next slide. So yeah, this one is just kind of showing some of the proposed units. I wanted to bring you guys attention to the red unit here, which is the pre-existing unit called the Papala unit, and it's been heavily outplanted already. So this is one unit that we are actually able to find some species data that was taken before they fenced it or before they started planting in it. So yeah, next slide, please. So yeah, we were able to get all of these data points for this unit, which is really helpful because, as you can imagine, if you're trying to survey for remnant species and you have a bunch of native species already planted, it might be a little confusing. So luckily we were able to get this file, and it kind of saved us some of that headache. I'm sure we'll still have to do some ground truthing in here as well, but that is one big relief for the project. Next slide, please. So yeah, kind of moving forward, we're really planning to take all this information from the ground surveys and the historical plants that we've identified that were here, and do some GIS analysis just to kind of gauge their resilience under future climate conditions, such as the droughts and all of that. It's an aspect that I haven't really gotten into yet, but this summer I'm hoping to really spearhead that. Next slide, please. So yeah, there's been some work already on like the vulnerability of species, but yeah, again, the goal with this project is kind of to focus in on of course pu'uvava. Next slide, please. Which is really just here on the map. You can see it's more on the leeward side of the island, and it's getting this information will potentially be helpful for the broader leeward community. Next slide, please. So one big aspect of this project has been community involvement. Of course, it's a community-based subsistence forestry area, but yeah, just keeping the community updated and working with them and making sure everybody's on the same page is a real major goal for this project, and I just want to highlight that. Next slide, please. So yeah, moving forward, we're gonna again just to reiterate, the major goal is to just inform specifically this unit, but also the broader pu'u as well. But for this unit, it's cool because it's not limited to just these native species. It's also potentially cultural species and even agriculturally significant like fruit trees and that sort of thing. So it's exciting to see where the restoration will lead us in the future. But mahalo for listening. This is pretty much my last slide, but also I want to just give a big shout out to the Pacific Islands Climate Adaptation Science Center. I forgot to put the logo here, but they're funding this project, and yeah, also there are other partners such as DOFA and the USFS, and of course Akaka Foundation as well, and then of course the community members that have all been integral to this project. Yeah, mahalo nui kako. And I'll take any questions now.
Mahalo, David. Yes, if you have any questions, feel free to put them in the chat. We are kind of running a little behind schedule, so we're gonna have to move on to our next presenter. But again, mahalo, David, for that awesome presentation.
Mahalo.
Our next presenter is Danielle Bartz, who will be presenting her presentation entitled Hawaii's Hammerheads, Understanding Distributions, Nursery Habitats, and Population Trends Over Time. And I'll hand it to you, Danny.
Awesome, thank you. I see my formatting got a little messed up, so bear with me. But today, I'm Danny Bartz. I'm a third-year PhD student in the Marine Bio grad program at Manoa and in the Hawaii Cooperative Fishery Research Unit advised by Tim Grabowski. So yeah, today I'll be giving you a brief talk on Hawaii's hammerheads and how we're taking an interdisciplinary approach towards understanding distribution, nursery habitats, and population trends over time. Next slide, please. So as many of you already know, issues in conservation biology often lack adequate monitoring data, and this makes it very difficult for resource managers to make informed decisions. Currently, there's a lack of baseline data for many imperiled species. However, there is a wealth of knowledge, observations, and experiences within local communities that can be tapped into. Next, please. So my focal species is the scalloped hammerhead shark. They are endangered throughout the globe. Sharks are culturally and ecologically important, especially here in Hawaii. And there's been a perceived decline of scalloped hammerheads within the local fishing community in Hilo. Next, please. So my objectives for this study are to characterize hammerhead nursery habitats, understand species distribution and population dynamics, and use the knowledge of the community to hindcast population predictions in lieu of baseline monitoring data. Next, please. So I hope to do this with three main approaches, those being local ecological knowledge, next, acoustic telemetry, next, and environmental DNA. Next, please. So next, I think, yeah. On the local ecological knowledge front, preliminary data has shown that anglers and community members have perceived increases in rainstorms and sedimentation in the bay, Hilo Bay, blacktip shark presence in the last 20 years or so, and fishing pressure, especially since COVID. Next, please. And there have been perceived decreases in scalloped hammerheads in the past 20 or so years, fish populations, and bagasse, which was the organic material left over from the sugar plantations, clearing out of the bay in the last 20 years. Next, please. So with that, I've been talking to folks with memories ranging all the way back to the 1950s. Through currently. So this is showing a total of 18 semi-structured interviews with fishermen and community members. Next, please. They fish from a variety of methods, whether it's from shoreline or boat, and hook and line, spear fishing, or nets, throw nets. Next, please. Next, please. And wow, this got all messed up. But on the left is some of the target species that have come up in the semi-structured interviews. And on the right, it's supposed to say bycatch species over there, sorry. That is some of the bycatch species. Next, please. With an emphasis on the oceanic blacktip. Now that shark that's pictured is a blacktip reef shark, but we're just gonna pretend it's an oceanic blacktip for today. But those keep coming up repeatedly in the interviews that they're kind of a nuisance and they're always being caught. Next, please. So regardless of that, a whopping 87.5% of people, when asked if they've ever seen or caught a hammerhead shark in Hilo Bay, said yes, with only a few people saying no or only having experiences on the Kona side. Next, please. And these are some of the areas within Hilo Bay that have shown up in the interview data as places where people have seen hammerhead presence. And this also includes 7 accounts from social media. So it's a sample size of 25 here. Next, please. So on this map, you can just visualize that last pie chart, and Bayfront North and Isles were the sites that had the most hits of hammerhead sightings or catch in the semi-structured interview data. So those are bigger. Next, please. And as you can see from this image from the National Ocean Service, the benthic habitat of Hilo Bay is mostly mud, so we don't have a whole lot of heterogeneity there. Next, please. So while the local ecological knowledge stuff is still underway, and I'm still continuing to conduct interviews with fishermen and work with community partners, the acoustic telemetry aspect of this is just getting kicked off. So after taking a couple of years to get all of our necessary permits, we finally have those. We finally have the supplies we need, moorings, tags, transmitters, and receivers, and our new boat. And the plan for this summer is to deploy the receiver moorings, tag and track juvenile scalloped hammerheads, hopefully a sample size of 30, and fish transects of the bay, because as you may have noticed from the interview data, it's obviously biased to the areas that people are fishing. It's the areas that they've caught sharks. So I'm trying to work with that bias there. Next, please. So while we've been waiting for permits and such, I've been dabbling in some environmental DNA. So I've decided to follow the methods from this paper by Alyssa Budd et al. That was published last year in Guam, where they were able to find the first detection of scalloped hammerheads in this one harbor in 5 decades. Next, please. And this is kind of how they did it with sample collection. They filtered the water, did eDNA extraction, and then species detection using qPCR. And then they added an additional step of doing bidirectional Sanger sequencing on any positive reads for confirmation. Next. And so my methods adapted from those methods are very similar. These just show some of the different equipment and supplies that I'm using, but following the same flow of sample collection, eDNA extraction, some cleanup there for inhibitors, and then qPCR and bidirectional Sanger sequencing. Next, please. So what this looks like is on the left, we have the filter cartridges that have a snorkel that they attach to, and on the right, we have the 5-micron nylon net filters. Next. Those get inserted into the filter and then attached to this battery operated diaphragm pump. Next. And then once the water is filtered, the filter gets removed and stuck in one of these tubes on the left that are filled with a lysis buffer and homogenization beads. Next. Sorry, there's a lot of pictures on this one, so you can just click through. So these are some of the 18 sites that I sampled on the Big Island. There's 2 more, I think. Thank you. And yeah, that's good. So those are some of the 18 sites that have been sampled so far on the Big Island. Next. And on the left, we have a Laea Bay on Maui, and then on the top right, Pearl Harbor on Oahu, and the bottom right, Nawiliwili on Kauai. Next. And then next, Hanalei on Kauai. So next. Now sampled a total of 30 sites within 20 different embayments throughout the main Hawaiian Islands. And what this basically looks like is collecting buckets of seawater next, whether it be from shore or boat, and then running it through the battery powered diaphragm pump, like I said, and then sending those filters off to the lab. Next. So yeah, that just shows me like chopping up the filter, putting it in the tube, and on the right, filtering the water. Next. So yeah, this shows the sites that I've done so far, and I think this is all the sites that I'm going to stick with, but I want to hopefully, if funding permits, redo this all in August, when would be kind of the more tail end of their supposed pupping season, and testing all of these waters for hammerhead shark DNA, if I didn't make that clear. So there's a total of 30 sites within 20 embayments throughout the 4 main Hawaiian Islands. Next. So currently those samples are at the Microbial Genomics and Analytical Laboratory at UH Manoa, and they're doing the DNA extraction and qPCR. And then, like I mentioned, any positive reads will be sent off to the ASGPB lab for bidirectional Sanger sequencing, and that's for confirmation purposes. Next. So what I hope to do with all of this data is take the local ecological knowledge, acoustic telemetry, and then the presence absence from the eDNA work and incorporate that into species distribution models, ArcGIS story maps, and habitat suitability models, so that we can have a better idea of population dynamics and species distribution, and also hopefully some nice story maps to share back with our community partners. Next. With the last part, the eDNA part of that, we're hoping to predict occupancy using landscape variables as proxies. So different variables that could influence hammerhead presence or absence within an embayment, such as population density in the Ahupua'a, land use in the watershed, benthic substrate composition of the bay, size and shape of the bay, and then proportions open to the ocean or of each benthic substrate. Next, please. Yeah, and those are my references. Next. And huge mahalo to DAR for some funding and in-kind support, Dr. Budd for her guidance on my sampling and methods, MGAL and ASGPB labs, all of my friends and colleagues that have helped with the sampling, and of course the anglers and community members that have graciously shared their mana'o with me. Next, and if you have any questions or you'd like to schedule an interview, you can contact me here. All right, thank you very much.
yeah, like Danny said, the chat is open for any questions. And it seems like we got a first question from Lillian. And they say, hi, Danny, sounds like a great dissertation. Is hammerhead presence in our nearshore waters seasonal? Say more of them during pupping season? If so, is your eDNA sampling stratified temporally or at any of your many locations?
Yeah, thanks, Lillian. Great question. Yeah, so it is a seasonal thing, and it's supposedly through like May through September is their pupping season. So you know, I had to do all the sampling before May because it was kind of just a funding deadline type of thing. But I'm hoping to repeat my 30 sites again in, say, August, because that would be kind of at the tail end of their season, and so I can add a temporal aspect to it.
And then John also has a question, and he says, have you had any success with detection of hammerheads in the eDNA samples?
So not yet. The samples are currently at the lab undergoing all their analyses, so I don't have that data yet, but the paper that I'm following and the methods that I'm following, I am faithful because they were able to detect hammerheads for the first time in 5 decades in that harbor in Guam. And they're following a different approach. So it's not just metabarcoding where you have to deal with a lot of like inhibitors and false positives and things like that, but it's a very species specific approach that was really kind of nailed down in this paper. So it's also, my project is kind of a proof for them, see if it works. Thank you for that question.
It seems like Steph also has a question, but we're going to have to go ahead and move forward since we're running a little bit late. But if
No
Danny, if
worries.
you want to, can you answer it in the chat?
Absolutely, will do.
And then again, mahalo, Danny. Our next presenter, our last presenter is Skyler Chong with their presentation entitled Recruitment Dynamics of Invasive Mullet Species in a Native Hawaiian Fish Pond. And I'll hand it to you, Skyler.
Thank you. Yeah, hi, my name is Skyler Chong. That's my project title. This project was done through the PIPES program last summer, but then I continued to do this project through till now. We're still working on it and are trying to publish this work in the future. Yeah, next slide, please. So my internship was at the Kumuola Marine Science Education Center. It's great because you all, I think, will be going there for the closing of this symposium, which is really awesome. You get to meet everyone there. It's a great crew of people. You can ask them any questions that you have that I don't get to today. But for those of you in Hilo or from Hilo, you probably have seen this site or passed by it. It's out in Keaukaha on the Maka side of the road from 4 miles or Peve. And yeah, Kumuola manages 3 local ʻŌhiʻa in this area, Waiahole, Kapalaho, and Waiopio. They function mainly as an educational site. They run through Kamehameha schools. Yeah, I'm excited for you all to get to go down there. It'll be great. Yeah, next slide. So for those of you who don't know local ʻŌhiʻa or fish ponds, these spaces were very common in Hawaii prior to Western contact. At that point, they were capable of producing 300 to 500 pounds of fish per acre per year. But due to colonization, they sort of often, or a lot of them have fallen to sort of disrepair. But since the 70s, a lot of effort has been put into rehabilitating and revitalizing these spaces. Efforts with local ʻŌhiʻa tend to hit on a lot of cool issues, food sovereignty, cultural revitalization, as well as ecological restoration, which we're focusing on today with TCBES. And so there's a lot of cool work going on there. I'm really stoked on it. I hope you all will be too when you go down there. Oh, can go back, please. Sorry. There are 6 different kinds of local ʻŌhiʻa. The main sort of stereotypical one that we think of when we hear the words local ʻŌhiʻa is the local kuapā, known as such because of its kuapā, its wall that extends out into the ocean, encapsulates a portion of the shore. Within the local ʻŌhiʻa, you get these brackish water environment due to the punawai, the freshwater springs. And this brackish water creates a really high it's a highly productive ecosystem, and a lot of fish, or a lot of our native species are drawn to it because there are a lot of resources in those areas. So their fish are wanting to get into the ponds, and they do so through these things called makaha, or sluice gates. They're basically gates in the wall that allow for small fish to come in. The fish then enter the pond, consume the resources there, and get bigger to a point where they can no longer exit. So it's important to note that these spaces are naturally stocking. In an ideal local ʻŌhiʻa, you're not adding fish, they're coming in on their own. Yeah, next slide. There are a lot of different species of ʻŌhiʻa in local ʻŌhiʻa, but the two that I'm focusing on today are the ama and the kanda. The ama is a species of mullet that is extremely important to local ʻŌhiʻa. It's a native fish. It's very culturally important. Generally, it's a very prized fish within our fisheries. And so in the objective of restoring a local ʻŌhiʻa, you're trying to have more ama in that space. It's like one of the key things, you know. In contrast, the kanda is also a species of mullet, but it's invasive. It's much smaller in size, generally less valued in our fisheries. And these two species are mullet, so it's thought to occupy the same ecological niche, consume the same resources, and potentially compete with each other. One important thing to note is that the ama spawns only once a year, while the kanda spawns year round. So just in terms of reproductive output, the kanda has sort of an upper hand in this. And that's why since the 1950s, when it was introduced, it has really skyrocketed in its populations. Yeah, next slide. So yeah, my work at Kumuola, I don't know why I say it like that, was sort of centered around this question of the relationship between ama and kanda, because again, the ama are so important and the kanda are this invasive species. And so we wanted to look at when are kanda coming into the local ʻŌhiʻa, and are there any environmental factors that we can use to sort of predict when kanda are coming in? Because if we can do that, then potentially we can come up with different methods to control the recruitment. And so to do that, you can ask them more about it when you're down at Kumuola, but there's this very specific context or situation there that allows for them to count all the fish that are coming into the pond and to genetically sample them to see which species there are. So they have a bunch of data starting in 2018 on fish recruits into the local and which species are coming in when. And so for my project, I was mainly looking at kanda, but in the context of this relationship between kanda and ama. Next slide. So this is, I'm sorry if it's cut off for you as well. The bottom is over time. So this spans from starting in 2018 all the way through 2021. But this is just the observed kanda recruitment in the ponds. You can see that it's not uniform. There are these huge spikes and then periods where there's almost no recruitment. And so my goal, my task was to sort of take this and say, when are those spikes and when are they happening and what can we tell from that? So what I did, next slide, was I took that data and I created a function that basically identified when those spikes were based off of the moving standard deviation and moving average. But basically it flagged all those days when kanda recruitment was significantly higher than usual. And so all those vertical lines are what I've called spikes. And so with that, you can sort of think about when are those spikes happening and what does that tell us? And so I decided to look at it through sort of two different lenses. Next slide. Oh, that's not the slide that I was going to have. But yeah, two different lenses, one of them being the Hawaiian lunar cycle. So the Hawaiian lunar cycle is split, the Hawaiian month is split up into three different phases: Ho'onui, Poipoi, and Ho'emi. Each are 10 days, and they generally correspond to the waxing, full, and waning moon phases. And then I also wanted to look at it in terms of season. So summer months and winter months, and sort of comparing those two. So each of these dots, this is kind of a confusing graph to just look at, but each of those dots corresponds to one of the vertical lines in the prior graph. So each one of those is a spike. And we can look at that in terms of season and this lunar cycle. And what this tells us basically is those clustering of dots in the top left corner means that there's increased condo recruitment in the winter, in the summer months, and during the Ho'onui and Poi Poi moon phases. So that just gives us an insight into the condo recruitment dynamics. Next slide. We can also just look at the average daily condo recruitment for each of these things. So for the Honolulu, you can see here that this is in line with what the previous graph showed, but basically recruitment is much lower on average in the Ho'emi moon phase, which is the waning moon phase. Yeah, and then next slide. And then also the condo recruitment is significantly higher in the summer months. So these are designated as May through October, and then winter was November through April. That's based on some other studies done on reef fish recruitment in Hawaii. But yeah, again, you see that recruitment is much lower in the winter, and that graph with the dots was sort of the combination of those two. So we can say that condo recruitment is higher in Ho'onui and Poi Poi during the summer. Yeah, next slide. Yeah, sort of in conclusion, just to wrap things up, this study gives insight into the life cycle of the conda, which in our local ʻŌhiʻa is existing in relationship to the ʻama-ʻama, very important species. So it's important as well to understand the life history of the conda, because it gives us insight into how we can take care of these spaces. Yeah, next slide. I think it's just, thank you. Yeah.
Thank you, Skyler, for that awesome presentation. And yes, the chat is open for any questions that you may have for Skyler.
Yeah, there are, sorry, I'm just reading the question. Are there similar daily recruitment data for the ʻama-ʻama so we can compare periods of peak recruitment? Yes, there is a lot of data on that for those same exact time periods. The genetic sampling didn't start until 2019. So, and it's a little hard to tell, and the methodologies changed a little bit. But yes, there is the data for the ʻama-ʻama. My project was just focusing on the conduct because they hadn't sort of run all these analyses on them yet. But there is recruitment data for the ʻama-ʻama. Generally, in recent years, in the past 2 years, in 2021, 2022, we're not seeing much recruitment, which is not great for ʻama-ʻama. But yeah. Kind of have increased spawning in the summer. That's a good question. It's hard to tell. This project is looking at when conda are recruiting into the pond, and they spawn in the deep ocean and then come into the coastal environments. And so there's a sort of a 2-month period from when they're spawning to when they're coming into the ponds. And because sort of summer is spanning this like 6-month period, it's hard to tell if they're spawning in the summer or if that includes the 2 months before sort of what we've designated as summer. But again, it's like really hard to measure spawning, like the actual spawning of conda, because it's happening in these environments that are really hard to get to, and it's really hard to measure. So it's actually a really cool opportunity to sort of get them as they're coming into the ecosystems and count them then, but it doesn't give us so much insight into their spawning just when they're recruiting. Yeah.
Well, thank you, Tyler, for that awesome presentation. And thanks to the audience for those questions. And yeah, this marks the end of the first day of the symposium. On behalf of the Kaiame'alo Club, the TCBES program, the La Honua Earth Day Committee, and all the other community partners who helped make today happen, we'd like to offer a big mahalo to all our presenters and a big round of applause. Yes. Also, thank you to the audience members who joined us today. We will be back tomorrow, April 29th at 9 a.m. for our keynote speaker with Lokelani Brandt as well as our final presentation session followed by awards. The TCBES annual silent auction is also currently open for bids, and we will close tomorrow at 3 p.m. So be sure to check it out. We will also be closing the TCBES Symposium with a workday at Kumuola Marine Science Education Center tomorrow at 1 p.m. There's still time to sign up, and we hope to see you there. Again, mahalo for a great first day at the TCBES Symposium, and we hope to see you there tomorrow. Aloha.